Economic importance of bacteria. You know that the bacteria plays a very important role in many ways in our day-to-day life. Now let us see whether they are our friends or they are our enemies, and it is up to you to justify it. Bacteria useful to mankind. Soil fertility. Nitrogen fixing bacteria. Nitrogen fixing bacteria are found in the soil either freely, that is, Azotobacter, Bisonichia, and Clostridium, or in root nodules of leguminous plants, that is, Rhizobium leguminosarum. Here is the Azotobacter free living, and nitrifying bacteria, the decay of plant and animal proteins affected by saprophytic soil bacteria returns nitrogen to the soil in the form of ammonia, and this process is termed as ammonification. Nitrification occurs in two steps, Nitrosomonas and Nitrococcus. You know, you have studied earlier while discussing the respiration that one converts ammonium compounds to nitrites, and then these nitrites are converted to nitrates, and plants they take the nitrogen supply in the form of nitrates. Decomposers. Decay of dead plants and animals. Some bacteria, for example, Bacillus mycoides, Bacillus ramosus, Bacillus vulgaris, etc., attack dead bodies of plants and animals and convert their complex organic compounds like proteins into amino acids and then amino acids into ammonia. This is Bacillus mycoides. The decomposition of dead plant and animal bodies largely by the bacterial action returns carbon dioxide to the atmosphere and nitrate to the soil. Aerobacter aerogens decomposes DDT, Flavobacterium and Corynebacterium decomposes 2,4-D. This is Aerobacter aerogens. Decomposition of petroleum on ocean beach. In recent years, a genetically engineered bacteria named as superbug, its technical name is Pseudomonas putida, was developed by Dr. Chakravarty, which can feed on oil spills and petroleum and thus proves to be of immense utility in curbing the water pollution of sea and oceans. And this is the superbug created by Dr. Chakravarty. Disposal of sewage. Decomposition of sewage waste, methane, and carbon dioxide are produced by methanogenic bacteria, for example, Mycrococcus and Methanobacterium. The methane is used as biogas, as fuel. This is Methanobacterium. In dairy, following bacteria are responsible for dairy products: curd, Streptococcus lactis; cheese, Lactobacillus lactis; butter, Streptococcus lactis; yogurt, Lactobacillus vulgaricus; buttermilk, Streptococcus lactis and Streptococcus cremoris. This is Lactobacillus vulgaricus, and this is Lactobacillus lactis. Industrial value. In vinegar industry, initially the carbohydrates undergo fermentation to be converted into ethyl alcohol by the action of the yeast. In the second step, the aerobic bacteria Acetobacter aceti and Mycoderma aceti oxidizes the alcohol to acetic acid. And here is the equation that how ethyl alcohol is converted into acetic acid by Acetobacter aceti. Fiber rating. The fibers of jute, hemp, and flax are separated from fiber-yielding plants like Linum acetatisima, which is called as flax or sun, Cannabis sativa, hemp or putsun, and Corchorus capsularis, that is jute. This process is carried out by Clostridium butyricum, Clostridium falcineum, and Clostridium pectinovorum. etc., inhibiting the water. This is Clostridium butyricum bacteria. In tobacco industry, Bacillus megatherium and Mycrococcus candidans is used for developing flavor and taste in tobacco leaves. In tea industry, by fermentative action of Mycrococcus candidans, the curing of the tea leaf is done. This is Bacillus megatherium and this is Mycrococcus candidans. Antibiotics. Most of the broad spectrum antibiotics are obtained from bacteria. Streptomycin from Streptomyces gracius, chloromycetin, which is called as chloramphenicol, from Streptomyces venezuelae, aureomycin, that is called chlorotetracycline, Streptomyces aureofaciens, terramycin, which is called as oxytetracycline, Streptomyces remosus, neomycins from Streptomyces ferredi. Erythromycin from S or Streptomyces erythraeus, kanamycin from Streptomyces kanamycetius, and bacitracin, Bacillus subtilis, polymyxin from Bacillus polymixa. Some disease-producing bacteria in humans. Diplococcus pneumoniae, pneumonia, Clostridium tetani, tetanus or lockjaw, Clostridium botulinum, botulism of food poisoning, Eberthella typhosa or Salmonella typhi, typhoid fever, Corynebacterium diphtheria. Mycobacterium tuberculosis, TB or tuberculosis, Neisseria meningitis, Neisseria gonorrhoeae, gonorrhea, vernal disease, Haemophilus pertussis or Bordetella pertussis, whooping cough or you call it as pertussis, Vibrio cholera, Pastorella pestis or Yersinia pestis plague, Treponema pallidum, syphilis, again VD. Shigella dysentery or Bacillus dysentery. It is bacterial dysentery, also known as shigellosis. Salmonella typhimurium, bacterial food poisoning. Mycobacterium leprae causing leprosy. Staphylococcus aureus, fever, lowered blood pressure, vomiting and diarrhea. Plant diseases, short rot of turnip, Erwinia cartovora, bacterial blight of rice, Xanthomonas oryzae, citrus canker, Xanthomonas citrae, crown gall, Agrobacterium tumefaciens. You know this bacteria is extensively used in genetic engineering. Toxins and the pathogen and the mode of action. Neurotoxin. Botulinum toxin, Clostridium botulinum is the source, binds to the motor neurons and prevents release of the neurotransmitter. Tetanus toxin, Clostridium tetani is the source, binds to the nerve cells and blocks inhibitory impulse transmission. Cytotoxins, diphtheria toxin from Corynebacterium diphtheri, penetrates the host cells and stops protein synthesis. Then enterotoxins, cholera toxin, source is Vibrio cholerae, stimulates abnormal pumping of electrolytes and water into the colon. Shiga toxin, Shigella dysentery, stops protein synthesis in the cells by interfering with the 60S ribosomal subunit. General characters, occurrence. Now we take up the cyanobacteria or blue-green algae are one of the most successful groups of prokaryotic, gram-negative photoautotrophic organisms distributed all over the world. Some cyanobacteria grow in most unusual habitats. A few species grow in hot water springs having a temperature range from between 70 to 75 degrees. Example is Formidium, and others grow at a low temperature in the polar region. Example is Nostoc. The red color of the sea is due to the abundance of the planktonic cyanobacteria, that is Trichodesmium erythraeum. This is Formidium living in the hot spring, and this is the Trichodesmium erythraeum, causing the red color to the sea. Cell structure, occurrence. The cell wall is firm and rigid. It is made up of peptidoglycan, similar to the bacterial cell wall. Inner contents of the cell can be distinguished into two regions, outer pigmented region known as chromatoplasm and central hyaline as centroplasm. The membrane-bound structures such as true mitochondria, chloroplast, endoplasmic reticulum, Golgi bodies, true vacuoles, etc. are absent. The photosynthetic pigment present in the cells are chlorophyll A, B, carotene, myxoxanthophyll, myxoxanthin, C-phycocyanine, and C-phycoerythraeum. This is the structure, a typical structure of a cyanobacteria, which is a prokaryotic cell. You can see here you have the ribosome, lipid droplet, cyanophysin granules, the so-called nucleoid or chromosomes, peptidoglycan cell wall, thylakoid membranes, starch grains, etc. The central nucleoid has several irregularly arranged fine strands of DNA. These DNA strands are naked, and that is, they are not associated with histone proteins. Some cyanobacteria possesses a special type of cells called heterocysts to perform a special function. They are present, for example, in nostoc, anabina, cytonema, and they are the sites of nitrogen fixation. You can see the anabina with intercalary heterocyst, the stick-walled structure. Vegetative reproduction occurs by fission, fragmentation, and by formation of hormogonia. Asexual reproduction occurs by the formation of echinids, endospores, and nanocytes. And that is, you can see these are the echinids, and in between this, there is intercalary heterocyst. And this is the formation of the hormogonia. There is no sexual reproduction, but genetic recombination has been reported in some genera. That is, for example, Cylindrospermum and Anabina species, etc. Ability of cyanobacteria to fix atmospheric nitrogen, particularly in wetland condition as in rice fields, is a highly beneficial phenomena in nature. This is the Cylindrospermum species where the genetic recombination has been observed, as you have seen in bacteria. Spirulina is being used as a source of protein-rich supplement to diet of human and also aquatic life. It is cultured, dried, powdered, and then supplied in the form of 1 gram tablets. This process is known as production of single-cell protein, in short SCP. Balls of nostoc commune are collected, boiled, and consumed as food by Chinese. The prepared food is called a youchou. This is the spirulina, which of course you can get in the market as a protein supplement. And this is the balls of the nostoc. Some species of Anabina and Olosyra do not allow mosquito larvae to grow. These species can be inoculated in ponds to prevent the growth of mosquitoes. Harmful activities. Mycrococcus, Anabina, etc. releases toxins in the surrounding water, which is harmful to fishes and other aquatic fauna. This is the Mycrococcus causing the water bloom. This is the water bloom. Excessive growth of the alga resulting in the death of the fishes.